Final stage of the cellular respiration or aerobic respiration, and it's called the electron transport chain. So this occurs on the cristae, okay? If I were to draw a mitochondria up here, right? So we have a circle, okay? And then we have the squiggly things. This electron transport chain is happening on this line, which is called the cristae, okay? And the cristae is a phospholipid bilayer. As I drew it right here, it's made of phosphates, right? And on top of it, it is the intermembrane space up here, and below it is the matrix. And then all of these complexes are on the cristae. So right here, we see a complex labeled 1, Roman numeral 1, 2, 3, 4, and then we have a protein called ATP synthase. So all throughout the cycle, all throughout cellular respiration, we made products, right? We made NADH and we made FADH2. Now, those FADH2 are going to come to the electron transport chain, and right now let's talk about NADH. So the NADH, and we made NADH in, as you can see through this graph, we made NADH in glycolysis, the precursor, and the Krebs cycle. So all of those NADH are going to come, and with the presence, the whole driving force of the electron transport chain is oxygen right here. This is the driving force, okay? And so because oxygen is present here, oxygen is it's the driving force of cellular respiration. So the NADH, only in the presence of oxygen, will be broken down into NAD plus and hydrogen ions. And it's also going to be stripped of its electrons. These electrons are going to travel into this complex number 1, and it's going to travel to complex number 3, okay? And then from there, these electrons are going to travel to complex number 4, where they meet hydrogen ions or protons that are in the matrix, because this area is the matrix, and they're going to combine with the electrons and combine with oxygen and make water, H2O. And so this NADH, every time the electron moves from here to here, it is releasing energy. So when this electron moves from here to here, it is releasing energy. And what does this energy do? This energy allows for this hydrogen ion to be pumped against its gradient and through the cristae. It's against its gradient because up here in the intermembrane space, it's a high concentration of hydrogens, whereas in the matrix, it's a low concentration of hydrogen ions, right? And it always goes from high to low, right? So when it's going from low to high, it needs some energy. It needs to be facilitated in order to be pumped against its gradient. And that's from the electrons moving and releasing energy, okay? So these hydrogen ions are being pumped through the intermembrane space, and then this NAD plus is going to be sent back to glycolysis and repeat the whole process. And then the FADH when we made FADH in the Krebs cycle, the FADH meets at, this NADH, it is providing a lot of energy, right? Let me talk about that later. So the FADH2, also gets broken down into FAD plus and hydrogen ions. These hydrogen ions are going to get pumped through its gradient, and this electron is going to be pumped to complex number 3 and then complex number 4, where again the electrons are going to meet oxygen and make water, okay? And this whole process, every time it moves from here to here, and there's also multiple steps happening. There's like complex here and here, but you don't have to know that. Every time it is being pumped, it's releasing energy, correct? And then this energy is pumping the hydrogen ions here. And this energy is enough. This energy is actually going to cause these hydrogen ions to all move to this area called the ATP synthase. And recall how I said that the intermembrane space has a high concentration of hydrogens, whereas the matrix has a low concentration of hydrogens. And as we know, hydrogens or anything always goes from high to low. So all these hydrogen ions that were pumped want to go through this ATP synthase and go back into the matrix. And this is going to be through the protein called ATP synthase. What ATP synthase does is that when the hydrogen ions go through it, it provides energy. And this energy causes this turbine, it spins in a motion like this and this and this. And that brings the ADP and inorganic phosphates that are found in the matrix so close together that they make ATP. This is basically this is the product. And this is what our cells need, correct? Because the whole reason we go through aerobic respiration or the cellular respiration is to make ATP for our bodies to use. And so the creation of ATP, it all stems back to electrons, right? Because the electrons are moving and it's giving off energy that pumps the hydrogens, right? So when NADH is pumped, it provides enough energy to make 3 ATP. Okay, this NADH being broken down provides enough energy for enough hydrogen ions to go through and make 3 ATP. And for every 1 NADH makes 3 ATP. But sorry for the intrusion, but on the other hand, because FADH2, it can't come to complex number 1 and be broken down. It has to be broken down from complex number 2, which means that these electrons that are broken down from FADH2, they have a shorter span, right? They're shorter in time in the ETC. And it provides naturally, if these electrons are only traveling this distance, but these electrons are traveling this distance, naturally these electrons are going to provide less energy, right? So then these electrons are going to provide only enough energy for 2 ATP to be made, to be made. So for every 1 FADH2 is 2 ATP. That's the electron transport chain. So our inputs for the electron transport chain are naturally NADH, which were made, all over. We made it in glycolysis, we made it in precursor, and the Krebs cycle. We have FADH2, which we made in the Krebs cycle. And our outputs are water, H2O, and then ATP. So if we go over this chart I made, and this is like a little review, okay? So in glycolysis, how many ATP did we make? 2. In glycolysis, how many in precursor steps, how many ATP did we make? 0. In the Krebs cycle, how many ATP did we make? 2. And if you want to figure out how all of that is, go watch my other videos. So if we were to add up, 2 plus 2 is 4, right? So in total, we make 4 ATP just through these steps, right? And in the ETC, there's not really any ATP made in the ETC because, they're all made at the end, right? In glycolysis, we made 2 NADH. In the precursor steps, we also made 2 NADH. And the Krebs cycle, we made 6. So in total, this is 10 NADH, right? And I said that for every 1 NADH is 3 ATP. So if there's 10 NADH, we have 30 ATP. Same thing for FADH. We only made 2 FADH in the whole cycle, in the whole process. So 2 times 2, because for every 1 FADH is 2 ATP, we make 4 ATP in total, right? So in total, what Miss Romana tells us is that 38 ATP are made. So in total, after cellular respiration happens, the equation for cellular respiration is C6H12O6 plus, remember, because we had to add 6 oxygen atoms, right here, oxygen, yields 6 CO2, 6 carbon dioxide, correct? Because we have 6 carbon dioxide right here, plus 6 H2O, which was up here, plus 38, or approximately 38 ATP. Or if it was an equation, you wouldn't write plus 38 ATP. You would just write plus energy. And that's your equation for for cellular respiration. So that's like pretty much the whole thing. There's not really anything else you have to go over, but if I were to go over anything, I'll just review everything for you. So in glycolysis, some things when I went to the our review session, some things I noticed is that people were confused why ATP is made from the NADH and stuff, because in glycolysis, ATP can only be synthesized after NADH is synthesized. And this is because NAD+, which is an electron carrier, has these specific traits that allows it to be broken down. It allows it to be broken, NAD+ has its specific characteristics that strips electrons from this compound, and it takes the electrons and hydrogen ions and it makes NADH, right? And so when this is stripped of its electrons and when it's broken down, these two phosphates are going to be added to the ADP found in the cytoplasm and make 2 ATP. And then the products of glycolysis is pyruvate, and in the Krebs cycle, what I noticed that people were confused that we have a compound named acetyl coenzyme A. Acetyl coenzyme A looks like this. It's a 2-carbon, and then attached with it is an enzyme or coenzyme A. Okay, so this coenzyme A is going to bring these 2-carbon into the Krebs cycle, where these 2-carbon are going to combine with these 4-carbon oxaloacetic acids, and it's going to make a 6-carbon citric acid. And then this coenzyme A is going to be released, and it's going to be recycled back into the precursor step, as we did again. It's a recycled thing. It constantly happens. Yeah, that's basically it, guys. Please do not be worried. Please tell me if you have any questions at all. Text me, call me, whatever you need. I'm here for you guys. That's it. I hope everyone does good, inshallah.